Alright, ladies, so in this video, I am going to introduce you to Chapter 12, and this is a little bit different approach than what we've done a lot of times. So rather than having the actual slides here, I'm just going to talk you through sort of on this Blackboard space, but to help you along with the first part of the notes for this chapter. Now, really, we're kind of merging chapters 12 and 13 here, but we're going back into genetics, but kind of from a different point of view. And the goal now is to look at things more at the molecular level. So we're going to try to understand DNA structure, and we're going to also try to understand how scientists sort of got us to this point of understanding. All right, so if we think back to chapter 11, we learned about Gregor Mendel, right? Father of modern genetics. And, Mendel taught us a whole lot. But, remember he was a priest. But, he was limited in what he could do for us. Because in his time, they really didn't have an option to understand the molecular side of all this. So the issue is that we've got to fast forward quite a bit to get to the point where we understand where your traits actually come from. Chapter 11 was all about the organism, right? Inheritance. You get this trait from your parents, but the question is, how do you do that, right? How does this trait of maybe it's dimples, right? Or maybe it's freckles. How do these traits actually find their way from your parents to your offspring? And it took quite a few scientists and quite a few years to figure that all out. So the first guy that we're going to talk about is a guy named Frederick Griffith. Now, Griffith was British, and Griffith was a microbiologist. So really, what he did is he studied bacteria, he studied infectious disease, and it was his goal to try and figure out how or why people get sick. Now, he had figured out, or he had sort of identified and isolated two different types of bacteria. In fact, these are specific type of bacteria called Staphylococcus, right? Or Staphylococci would be the plural of that. And he had found these staphylococcal bacteria in mice. Now, like I said, he found two types. One type he called smooth. And these smooth staphylococcal bacteria looked smooth. You'll see a picture in your textbook and in your notes of sort of this petri dish of growth. And when bacteria grow on a petri dish of this type, they form these little round, smooth colonies. And these were actually pathogenic. Right? And pathogenic meant that they caused disease. So, we'll kind of draw that dripping, right? Now, he also identified a second type of staphylococcal bacteria that we called rough, or he called rough bacteria. And that's because instead of nice little smooth colonies, these guys grew in these little kind of haphazardly shaped colonies. Now, the thing about rough is that it was not pathogenic. So rough Staphylococcus did not appear to cause any kind of infection or any kind of disease. All right, so what Griffith did is he took mice and injected them with these bacteria. So, you'd have one mouse here, kind of draw it like this, right? And he would inject this mouse with the disease-causing strain, right? The smooth ones, which again, these are pathogenic. These should cause disease. And, how about this? You inject these smooth staphylococci into this mouse, they get pneumonia, and they die. Right? Now, he takes a different mouse. There we go. Right, and he injects into this mouse those rough bacteria. And again, these did not cause disease, so these are not pathogenic. And believe it or not, they were healthy. Right? They didn't get sick. So again, he identified these two types of bacteria. Some of them cause disease, some of them don't. Right? And that might be a new concept to you guys. A lot of times people just hear bacteria and you think bad, but there's really a lot of good bacteria out there that won't hurt you. Now, Griffith was really trying to figure out how or why these bad bacteria caused the disease. He didn't know if it was actually like a chemical that they were producing, or if the cells themselves were just interacting with our cells. So he tries another thing. So he takes a mouse, right? And now what he injects into this mouse is basically dead, smooth ones. They were heat-killed bacteria. So heat-killed, smooth bacteria. Now again, if these are dead, you would kind of think they wouldn't do anything. And sure enough, that's what he found. All right, so even if you use these smooth ones, which are pathogenic and cause disease, they don't cause disease if they are already dead. Now then Griffith moved on to one more idea. And this was really just sort of almost out of curiosity more than anything, that he wanted to try something else, sort of an extension of a control of sorts. And what he did is he took the rough bacteria, which again did not cause disease, so not pathogenic, and he added with that these heat-killed bacteria, so dead, smooth, which also did not cause disease. But sure enough, when he injected a combination of harmless bacteria and dead harmful bacteria, these mice got pneumonia and died. Now, this was baffling. When Griffith saw this, he wasn't really sure what to think. And he had a feeling that there was something else going on here, right? Now, what Griffith calls the something else is transformation. Because here's the thing. When he added the harmless bacteria and the dead harmful bacteria, and then he got this dead mouse, right? He also could culture from that mouse living harmful bacteria. So either these guys right here were finding a way to rise from the dead, right? So there's some sort of weird zombie bacteria. I don't even know how to draw a zombie. Or somehow these harmless ones were learning to be bad, right? And he called this transformation. The idea that a bacterium could actually change its DNA, or at this point we don't know it's DNA, but change what it does. Now, Griffith kind of left this alone. He, again, was more interested in infections and diseases and all that good stuff. And so he sort of left this up to the geneticists to start to figure out what it was that causes transformation. But here's the interesting question that comes from Griffith's work that a lot of scientists started to ask. Right? Basically, Griffith also talked about a factor that causes transformation. And so the question becomes, could Griffith's factor be the same thing as Mendel's factor? In other words, could the thing that changes these bacteria from one kind to another be the same teensy tiny little thing that allows organisms to pass their traits on to the next generation? Now we fast forward a little bit. Now we're talking about the 1940s. And we're actually going to be in the U.S. pretty much for the first time here, too, at New York University, where there is a man named Oswald Avery. He's a very small man. There's a fun little picture in your notes. He always wore these little spectacles, but he was a little dude, bald head. And Oswald Avery was a scientist who wanted to sort of further the work that Griffith was doing. So what he did is, instead of using actual bacteria, he used an extract of bacteria, which basically meant you take the bacterium and you break it up into its biomolecules that we learned earlier this year. So carbs, proteins, lipids, and this thing that was kind of new in 1944 called nucleic acids that nobody really understood just yet. Now, what this allowed Avery to do is test each of these different factors and see which ones did or did not cause transformation of bacteria. So essentially, he did the same experiment that Griffith did. He injected mice with each of these things, or actually what he did is he injected mice with an extract that left out one of these things in each case. And he was building on the hypothesis that proteins were actually responsible for transformation, and so then potentially for inheritance, as Mendel had explained. So what he does, right, is he takes his first mouse, all right, so now we'll go with a Mickey Mouse looking head here, and he hits it with his first extract. And his first extract is going to exclude proteins. So basically, he injects this mouse with carbs, lipids, nucleic acids, but no protein. All right, and what he finds is that they still get transformation, and the mouse dies. So he tries something different. All right, now takes another mouse. He hits this one with proteins, lipids, nucleic acids, but no carbs. And sure enough, transformation happens. Mouse dies. So he goes on to his third approach. There's our mouse. Hits this one with carbs, proteins, nucleic acids, but no lipids. And sure enough, you still get transformation. Mouse still dies. So he goes to the last option here, the one that no one really thought was what we were going to be looking for. And he hits this mouse with carbs and proteins and lipids, but no nucleic acids. And without nucleic acids, you get no transformation. And the mice are fine. And so what Avery does with this experiment is showed that nucleic acid molecules are the molecules responsible for transformation. And if they are the molecules responsible for transformation, they are probably also the molecules responsible for inheritance, as Mendel explained it. Now, the next scientists to do some work in this area are actually a duo. A guy named Alfred Hershey and a woman, one of the first female scientists we've talked about, named Martha Chase. Now, Hershey and Chase were working in the U.S. in the 1950s, and they worked with an odd little thing called a bacteriophage. Now, a bacteriophage is something that in the 1950s was a brand new discovery. This was one of the smallest little things that scientists had been able to identify. And a bacteriophage is actually a virus, but not a virus that would infect like you and me. It doesn't affect humans or larger organisms. It's a virus that infects bacteria. So it's super duper tiny, right? And what you get is this little thing. There's a picture in your notes. Kind of has this odd structure. It sort of has this almost insect-like look to it. Has this sort of body part here. The capsule, little tube comes down, and it's almost got these little legs. Now, what this bacteriophage will do is it will land on bacteria and then infect them. In fact, what it does is kind of strange. It injects some sort of material into the bacterium, right? And once it injects this material into the bacterium, it's almost like it can kind of take over, right? And this bacterium becomes sort of viral. And what it does is it actually ends up making a whole bunch of these little bacteriophage things. So basic way that a virus works, we'll talk about this later this year, but this is happening at this bacteria level instead of like a virus infecting one of our cells. Now, the question, as you might guess, is what is this stuff that's being injected into these bacteria? And so what Hershey and Chase did is they hypothesized that DNA, or nucleic acids, are probably what are being injected into these bacteria by bacteriophages. And if this were true, it would further support the work that Avery had done to show that the nucleic acids, or the DNA, are the genetic material of inheritance. So, what did they do? This experiment is a little bit confusing sometimes to students, and I understand why. It's just sort of a complex idea. But basically, a bacteriophage is made up of two things. It's made up of protein and DNA, right? So basically you have this sort of like protein shell, right? So that's what all this purple stuff is. And then you have some DNA on the inside of that shell. All right, so again, the purple stuff is protein, green stuff here is DNA. Now, these are crazy tiny, so it's not like scientists could actually see this or anything. So what they did is they tagged or sort of radioactively labeled different things. In the first example here, we're going to radioactively label phosphorus because there's phosphorus in DNA, but not in proteins. Right? And then they're going to do a second little thing, second trial. They're going to take a phage. It's going to have this DNA inside. And they're going to label, this time, the protein. Right? In fact, the way they're going to do that is they're going to label it with radioactive sulfur, because sulfur is found in protein. Now, in both instances, then, they're going to allow these things to inject bacteria. So, put a little bacterium on both of these. And we're going to allow the bacteriophage to come in both instances. Right? And they're carrying this DNA inside. Right? But again, in this one up here, the DNA is labeled or tagged, and this one, the protein, is what's tagged. And here's what Hershey and Chase found. In this second trial, right, when all was said and done, the bacteria went their way, and the phage went its way, and all that labeled sulfur stayed with the bacteriophage. But in this other first trial, bacteria went their way, phages went the other, and all that radioactive phosphorus was actually found inside the bacterium. So in other words, they marked DNA so that they could actually trace it with some computers and stuff, and they found that DNA was transferred from the phage into the bacteria. So this gave us very definitive evidence that DNA is what is transferred between cells like this to change their traits. So that takes us to DNA, right? Stands for deoxyribonucleic acid. All right, we're going to find the DNA in these long strands, primarily in the nucleus if we're talking about a eukaryote like ours, right? And your DNA is like your blueprint for the cell, right? A blueprint that tells us like how to build something or how to make something like that, right? And so these blueprints for the cell are all the instructions, right? All of the instructions, and the instructions for how to do anything that cell is supposed to do. Right? So just like a new device, or like my kids' toys they get at Christmas, they come with these instructions right, on how to put it together and how it functions, how to use it. And that's what your DNA is. Right? Your DNA is this instruction manual for the cell on how to do everything the cell does. But DNA is actually made up of smaller things called nucleic acids, or sometimes we just refer to them as nucleotides, because that's a little bit easier. Now, every nucleotide has basically a 3-part structure. This is not going to be a very accurate drawing here. But every nucleotide has a deoxyribose sugar, right, which is a 5-carbon sugar. So this has 5 carbons. It's called deoxyribose. Then that deoxyribose has a phosphate group attached to it. It's a PO4, all right, but a phosphate group attached. And that's true of every nucleotide out there. But then the nucleotide has a third thing. And this third thing is called a nitrogenous or a nitrogen base. Now, the nitrogen base is what can be different between each nucleotide. And there are 4 possible nitrogen bases. All right, the first 2 are called the purines, and that is adenine and guanine. Now, these have a similar structure. Adenine and guanine are both, if we look at it from like a chemical standpoint, they are both 2-ring structures. But then we have another group called the pyrimidines. And those are cytosine and thymine. Now these guys have just a single ring structure from a chemistry standpoint. So they have a similar shape to them. Now, it's good to recognize these full names, but as we move forward, we pretty much always use the first letter to abbreviate them. So we've got A's and G's and C's and T's that we find in your DNA. Now, nucleotides are typically found in chains or groups that we refer to as strands of DNA. Now, a DNA strand has a backbone made of alternating sugar and phosphate. So essentially, we've got a phosphate that is then attached to a sugar, right? That is then attached to a phosphate, which is attached to a sugar, which is attached to a phosphate, which is attached to a sugar, and so on and so forth. I think you get it. Now, this is referred to, again, as sort of like our backbone of the DNA strand. Now, sticking out from this backbone are those third pieces of the nucleotide, the nitrogen bases. So the A's, the T's, the C's, and the G's. Now, these 4 bases can appear in your DNA in any sequence, any different combination that basically is what you've probably heard of before as your genetic code, because the sequence of bases codes for information. It's sort of like we put together groups of letters to form words to have a language, a communication piece. Same idea here. We're going to string together sequences of these nitrogen bases in particular order that will give us some information. Now, the question that scientists had to figure out is, what is this actual structure of DNA? Because we are totally talking about something right now that is submicroscopic, right? Not something that, particularly in the mid-1900s, we had the ability to actually look at under a microscope. Now, what scientists knew is that DNA, doesn't really show up as individual nucleotides, and it doesn't really show up as simple strings either. And we know this thanks to a really important but often sort of unsung scientist named Rosalind Franklin. Now, Rosalind Franklin was a young British female scientist who was an expert in something called X-ray diffraction. Now, X-ray diffraction is still around today. It's a technique that actually involves quite a bit of radiation. But X-ray diffraction was sort of a weird, obscure thing. Basically, you could put some sort of chemical into the machine, and you would get out a unique pattern or sort of a unique picture. Now, if you look in your notes, because it'll be way better than what I'm doing here, you see a picture of an image that Rosalind Franklin got when she did X-ray diffraction, and it looks a little bit something like this. And the reality is that unless you're trained in X-ray diffraction, which I am not, and I doubt you are, it's really difficult for us to interpret what we're looking at. But when Franklin saw this particular image that you see in your notes, she knew basically two things about DNA. She knew that it consisted of two strands, sort of like tied together, right? And she could tell you from this picture that it formed a helix or a helical structure. So you can think of like sort of a coiled up spring would be a good example of a helical structure. Right, now Franklin figured this out, and Franklin worked for or worked with a scientist named Maurice Wilkins. Now his name doesn't really matter moving forward, but she worked with a guy named Maurice Wilkins. And Maurice Wilkins had a couple of other friends in the scientific world, and this is where the story gets a little more interesting. Now, Wilkins was a guy who worked in some ways with two other scientists, a man named James Watson and a man named Francis Crick. Now, I'm going to guess that you guys maybe have heard of Watson and Crick before. In fact, if you've heard of anybody that I've talked about in this video, you maybe have heard of Watson and Crick. And Watson and Crick used basically Rosalind Franklin's information, along with work that they had done as well, to suggest that DNA has a double helix for its structure. So instead of just being a single strand, Watson and Crick suggest that you have one strand of DNA attached to a second strand of DNA. And these are combined together along their lengths by those nitrogen bases. Now, why does the story get interesting? Well, the reality is that Rosalind Franklin really didn't offer to give her information to Watson and Crick. And it kind of depends on who you believe in this story, but it sort of sounds like Watson and Crick kind of stole Franklin's work, which at the time we're talking about wouldn't have been surprising since she was a female scientist. And unfortunately, of course, they did win a Nobel Prize for figuring out the structure of DNA, and would you believe that the people who won the Nobel Prize were Watson and Crick and a guy named Maurice Wilkins, and they totally left Rosalind Franklin out. Now, at first people think, oh my gosh, they totally forgot about her. Well, not exactly. There's a couple different rules when it comes to Nobel Prizes, right? The first rule when it comes to a Nobel Prize is that you can't give it to any more than 3 people. So there's only 3 people that can be allowed to be named on a particular Nobel Prize. The other issue is that you cannot give Nobel Prizes posthumously, meaning you can't give it to people who have died. And by the time the Nobel Prize was given for the structure of DNA, Rosalind Franklin, unfortunately, had passed away. Now, we're still not done with the story, though, because Rosalind Franklin died at a young age, most likely due to radiation exposure. And if you caught it a minute ago, I told you that X-ray diffraction used a lot of radiation. So Rosalind Franklin probably sort of caused her own death by doing this scientific work, and then hardly gets credit for it. So we're going to do everything we can to remember that Rosalind Franklin really contributed a lot to the structure of DNA, and it wasn't just Watson and Crick and Wilkins that did that. Now, the DNA double helix, if we sort of untwist it, has basically those 2 backbones, right, with those nitrogen bases sticking out from those backbones. And what Watson and Crick did figure out is that these nitrogen bases in this backbone are held together by weak hydrogen bonds. It's basically a little attractive force that connects the nitrogen bases on one side of our DNA double helix to the nitrogen bases on the other side of the double helix. And what Watson and Crick found is that there was a predictable way in which these paired up. And they worked with another scientist who kind of, again, provided them with some information, but not in a bad way. The other scientists had published a paper that they were just aware of. But what they figured out is that if you look at a DNA double helix, if there's a C on this side, there's always a G on the other strand, and vice versa. So if there were a G here, there'd be a C over here. If there's an A on this side, then there's always a T on this one. And again, vice versa. If there was a T here, there'd be an A there. Now, we call this, right, the rules of base pairing, or AKA Chargaff's rules. And Erwin Chargaff was another American, although he doesn't really sound like it, who had discovered that when you looked at a DNA strand, right, you would always find that the percentage of A's was equal to the percentage of T's, and the percentage of C's was equal to the percentage of G's. Now, when Chargaff figured this out a little bit before Watson and Crick, he published this idea, but he didn't know exactly what it meant, right? He just found that, hey, look at this. There's always the same amount of A's and T's, and there's always the same amount of G's and C's. But what Watson and Crick looked at that and said is, okay, for every A, there is a T, and for every C, there is a G. And so it allowed them to make sense of what we now call the rules of base pairing. And so you got to make sure you memorize these rules, okay? You definitely have to know that G always goes with C and A always goes with T as we move forward in these next couple chapters. Okay, so for our last little piece here, let's get away from these scientists for a minute and let's talk instead about how DNA is organized in the cell, right? Now, you probably already know, and we've been using this word for a long time in our class, but DNA is organized as chromosomes. But what you maybe didn't realize is that a chromosome actually involves a lot of different sort of almost layers of organization to it. Now, there's a big difference when we start talking about genetic material between a prokaryote, like bacteria, and a eukaryote, like us. Right? Now, in a prokaryote, we see just a single and circular chromosome. So, that's a little odd, I think, for some people to think about, but there's only one chromosome in a bacterial cell, a prokaryote, and it's actually a loop. So it's still double-stranded, right? But it's almost kind of like they just have this circle of double-stranded DNA. It looks something like that. All right, now remember, these guys do not have a nucleus. So that DNA is just kind of loose inside the cell. And this is roughly about 3,000 genes. That's not much. All right, so we would say that they have a tiny genome. That's not a lot of DNA. Now, eukaryotes, we've got a lot greater complexity, right? In eukaryotes, we're talking about multiple and linear chromosomes. And so for us, we're talking about a whole bunch of chromosomes that have more of just a line shape, right, to them. Now, we do, again, have a nucleus. So you're going to find these chromosomes inside the nucleus of a cell. So remember, eukaryotic cell is a lot more complex, and we're going to find that genetic material, deep inside this nucleus. And then you can probably already tell me this, but don't forget that you've got 46 chromosomes in a human cell. And this represents a lot of DNA, probably about 1,000 times the DNA that we see in prokaryotes. Now, the next question is pretty wild here. If we think about humans, we literally have about 3 billion base pairs, or 3 billion nucleotides inside of a single cell. Right? And this represents, somewhere, just kind of depends on who you talk to, but somewhere probably on the order of about 100,000 different genes. So this is a little bit mind-blowing here, but that means that a microscopic cell in your body that could fit on the tip of your pencil contains about 1 meter of DNA. So think about what a meter stick looks like, right? Right, meter stick, and then you've got a tiny bacterial cell like this, boop, right, or a tiny human cell, smaller than that, right? You can't even see it with your eyes usually, and all that DNA is somehow inside that thing. This means that DNA has to be packed in a ridiculously efficient way, and the way that DNA is packed is in a sort of a combining with protein that we call chromatin. Now, basically what you can think about is sort of like wrapping a string around a spool. If you think about a spool of thread or something. This allows us to put a massive length of something really skinny, right? Think about your thread, into a tighter space by wrapping it around something. So chromatin is made of these proteins called histones. And what we do is we take this histone protein, right, and then we take our DNA and we wind it around and around and around and around and around and around and around that histone. Right? And then what we actually do is we take a group of these histones together, wrapped with all this DNA, and we call that whole thing a nucleosome. A lot of times we call these beads on a string. So basically you've got this DNA, this huge length of DNA that gets wrapped around these little histones to put a massive length into a much tighter space, right? A much smaller place here. And this is called a nucleosome. Now, we keep on going though too. The nucleosomes, and there's a good picture of this in your textbook and in your notes, the nucleosomes coil together to form what's called a thick fiber. And then a thick fiber, when it comes time for cell division, will do more coiling, we call it supercoiling, to form a chromosome. So what I always tell students is, if you think about it, we always draw a chromosome like an X, right? When we're drawing mitosis and whatever. But this is sort of like if you've ever, look at the floor in your bedroom and you think you see a bug, right? Because you see this little dark spot on the carpet. But then when you walk over to it, you realize that it's just a wadded up string from one of your sweaters. That's basically what a chromosome is. It's a really tightly wadded up group of DNA and protein that is so tightly wound that it almost starts to look solid and appear like an X. And so that's how chromosomes are structured, or chromatin is structured, to fit a crazy large amount of DNA into your teensy tiny nucleus. Now, I know that was a lot of stuff. Again, you might want to look at the first 2 sections of chapter 12 in your book to help support you with this as well. And then the next time we're together in class, we're going to march onward and start talking about how you actually use the DNA that's in your cells.